In the previous video, we have discussed about the plasticity of the cancer cells. That is, when the heterogeneous population of the cancer cells are exposed to the drug, and how they can reprogram themselves in a way that they become drug tolerant. And we have discussed different scenarios that if the patients are continuously exposed to the drug, then how they can become drug tolerant, or if the patients are not exposed to the drug, then how the cancer cells modify themselves. So in this video we will discuss how those plasticity of the cancer cells can be targeted using different strategies, and they have been categorized into these three different categories. And first one is about this preventing tumor cell plasticity, that is preventing the heterogeneous cancer cell populations to ever become drug tolerant cells. And this can be done based on the information that has already been known about the heterogeneous cancer cell populations, such as when the reprogramming occurs and the cells become drug tolerant, this chromatin modifying enzymes such as HDAC and KDM uh, related genes helps in this helps the this chromatin helps the drug tolerant cell, these heterogeneous cancer populations to become drug tolerant cells. So this chromatin modifying enzyme that are related to these genes HDAC and KDM can be targeted, and some of the transcription factors adaptations have also been observed in the drug tolerant cells from this heterogeneous cancer cell population, such as BRD4 transcription complex. They can also be targeted to prevent the heterogeneous cells populations to become drug tolerant. And the signal, moreover, apart from this, the signaling pathways that helps these heterogeneous cancer cell populations whenever they are exposed to the drugs to become drug tolerant, such as WNT pathway, STAT3 pathway, XR pathway, can also be targeted, or can also be uh, taken care of account along with this drug injection, so that the cells populations does not become drug tolerant. And finally, some of the cancer cells, some of the cells populations that are more prone to the tumor microenvironment, such as fibroblast and macrophages. And as we have discussed, the M2 macrophages has the capability for this tumor immunosuppression, and tumor proliferations can also be targeted using this some of the genes that have been identified, such as HGF, PDGF, TNF, IL6. These are about the first categories. And next one is about the targeting new cell fate. That is, sometimes it has been known that if some kind of drug, cancer cells are exposed to some kind of drug, then they might lead to uh, what kind of changes that might have occurred, that could have occurred to this heterogeneous cancer cell populations, and how they modify themselves to the drug tolerant cells. For example, when the heterogeneous cell populations are exposed to drugs, one of the common scenario that has been observed is that they modify themselves into the mesochemical cells type. That is, EMT transitions might have occurred using these AXL and GPX4 modulations, and we have discussed about how this EMT transition occurs because of this GPX4 in our previous videos, where we are discussing about ferroptosis. So, and how we discuss this, how the ferroptosis has dual role, that is to immune suppression and also to kill the cancer cells. So these things can be targeted whenever the cells are exposed to the drugs, to prevent the reprogramming within the heterogeneous cancer cell populations and become drug tolerant. So these are about the second categories. The third category is quite broad, where the idea is to reverse the cancer cell plasticity from being drug intolerant to become resensitized towards the drug. So in this case, some of the epigenetic factors may play a role that has been understood, or that has been studied, that these epigenetic factors are more commonly responsible for reprogramming of this heterogeneous cancer populations to drug tolerant cells, such as EGH2 REST. And also along with that, some of the reprogramming transcription factors along with the epigenetic regulators, transcription factors are also be targeted, such as SOX, BRN2, MIF2, MITF. And The EMT transitions because of the cytokines such as TGF beta, E-cadherin can also be controlled can also be controlled or inhibited at the time of drug exposures to prevent the transition of the heterogeneous cancer cell populations to the drug tolerant cells. So these are some of the strategies in brief how they can be uh, improved to target they can be improved and used along with the targeted cancer cell therapy to prevent the heterogeneous cancer cell populations to become drug tolerant cells. And in this case, and if these strategies are successful, then cancer cell patients might live a longer life and without any repeated cancers, without any repeated treatment, or the gap between two treatments might be increased significantly. So I hope you got an idea about how the strategies, what are the strategies that can be used along with this target cancer cell therapy to prevent the plasticity of the cancer cells. If you need to understand more about these strategies in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.